Hi, I'm Chun Young, and I'll be demonstrating telomere length prediction tool using WGS and clinical data through AI models. We see that there have been many methods that have been developed to measure the telomere length, either computationally and experimentally, and their studies have focused on finding out associations between telomere length from a small number of clinical items and its specific diseases. So no studies have been developed. No studies have attempted to create a model from a large number of items. This is because it is difficult to model biological relationship among a large number of clinical items. Addition of each clinical item requires additional consideration of relationship with other items, which has to be reanalyzed in the study. Since the steps require time and effort, we try to predict telomere lengths using deep learning or machine learning models. As a data status, we have whole genome sequencing data for 10,033 samples in total, and all samples exist with clinical data. To measure telomere length and carry out deep analysis, we need VCF, FASTQ, and BEM files. As for clinical data, we have 29 categories with 329 subcategories manually annotated, and 7,820 samples, all duplicates, and all diseases included as well. As for 7,800 samples, they can be measured programmatically. And within the samples, we have 1,768 samples that has been experimentally measured in the lab. Okay. So this is the general step of the project. As mentioned above, we need BEM files, EMR, and BCF files for GIS analysis. And as for computational measurement, we can use different tools such as Telomere Hunter, Telomere Cat, and Telseq. Which tool to use totally depends on its accuracy and variance, which will be constant, and these parameters will be constantly measured. And then when we get covariate files and variance, which will be when we get covariate files and processed EMR data from 10K clinical data, we put into the AI test models, of which specific model was yet to be determined. As for BCF files, we put into the GIS analysis to get all significant SNPs for analysis. Some part is missing here. We'll be using SNP data of certain p-values, p-value threshold to get genetic determinants. As for AI test models, we'll be using genotypes of all samples at location of significant SNP and telomere lengths as an input and test the model as well. Then we may combine the test models, and from the first part and second part as well, we divided models so that we can see how the model would work for each variables. So this is the general flow of my projects. And this is the telomere length measured computationally. Yes, and we can see there is a slight relationship between the age and the telomere length, but we're measuring the length again using different tools as well. And this is the specific steps for the first part, where we have to use the different tools to measure the telomere length computationally. I'll be specifically describe the steps in the next presentation. Thank you.